Welcome and good afternoon to today's session of Dietary Patterns in Health and Disease, Feeding Your Microbiota, our latest series from our professional development team. So just want to remind you about upcoming sessions. They're obviously up there now, and sessions will be recorded, and all recordings as well as handouts will be posted on our landing page. That the link is in the chat box. Next slide, please. Just a reminder about who our team is. Next slide, please, Dr. Archer. There we go. Here's our team. I'm Julie England. I'm FCS Agent Emeritus in Seminole County, Florida. Our team leader and speaker today is Dr. Wendy Dahl, Associate Professor for the UF IFAS Food Science and Nutrition Department. Also with us are Wendy Lynch, FCS Agent in Putnam County, Florida. Carlin Raffi, who's an Associate Professor and Extension Specialist at Virginia Tech. Julie Garden Robinson, who's probably still recovering from our NEA FCS conference, who is a professor and nutrition specialist at North Dakota State University, and Kendra Zamoyski, who is a regional specialized agent up in the northwest part of Florida. Next slide, please. Just a short disclaimer, the information that we provide is for educational purposes only, and any reference to a commercial product does not constitute an endorsement by our team members or their institutions. Next slide, please. And just a little bit of information at the end of the Qualtrics survey that'll be sent to you either later today or tomorrow morning. There is a certificate of attendance. We'll record everything. Our evaluations are short, so please fill them out. And if you like the session, please tell your coworkers there's registration information on our MailChimp landing page where we have all of our handouts and the recording, but there is also information on registration. So please share. All right. And next slide, please. Just a little bit of information for those of you who may be new to Zoom, which I doubt any of you are, but there is a chat box that you can click on. Please, you can either send your questions to panelists only or to all panelists and all attendees. So we encourage you to put in questions and we will answer questions at the end of the session. If you don't see your little box, you can kind of hover your mouse over the bottom of your screen and you should get controls that will pop up. And with that, I'm going to turn over to Dr. Wendy Dahl to introduce our speaker today, whose photo is on the next slide.
Looks like we moved a slide, sorry about that, but it is my pleasure to introduce Dr. Doug Archer. He received his BA degree in zoology and his MS degree in bacteriology from the University of Maine and his PhD in microbiology from the University of Maryland, where he conducted research in virology and immunology. Upon receiving his PhD, he joined the Commission Corps of the U.S. Public Health Service and held various positions in the U.S. Food and Drug Administration, the FDA, and also FAO and the World Health Organization, capping his career as Assistant Surgeon General and Deputy Director of the Center for Food Safety and Applied Nutrition. Upon retiring from the Public Health Service in late 1993, Doug Archer was appointed Professor and Chair, Food Science and Human Nutrition Department here at UF. He stepped down as chair on January 1st, 2001, and since he has served as Associate Dean for Research, Interim Dean for Research, and in 2016, he returned to the Food Science and Human Nutrition Department as a professor where he teaches the graduate courses Research Planning and Intestinal Permeability and shares responsibility for the Food Science Seminar. And so here's Doug.
Thanks, Wendy. Can you hear me?
We can.
Okay.
We can hear you.
Could you go back one slide? Somehow I can't go backwards. Well, okay. No, okay. Let me try now. No, for some reason I'm skipping to the second slide. That's okay. I'll just read it. The title slide that I had, which doesn't seem to want to come up, was called The Zoo Within Us, Introducing Your Gut Microbiota. And start out, it's a pleasure to be here, and thanks all for attending. And I will try to impart some knowledge without being too confusing, which is a real trick when you're talking about the gut microbiota. Introducing you to your gut microbiota with a 30-minute talk is a pretty tall task. Literally tens of thousands of journal articles have been written about the gut microbiota, and more recently about the gut microbiome. Even so, I'd suggest that with regards to our understanding of the size, scope, physiologic importance of the gut microbiota, we are about where astronomy was when the Hubble telescope was launched. In short, we ain't seen nothing yet. It's estimated that we humans contain between 300 and 1,000 different species of bacteria in our guts. But given that we can't grow an estimated 90% or more of those microbes present, we're probably underestimating. And the microbiota is not just comprised of bacteria, as there are other critters in our intestinal zoo, such as viruses, in fact, as many viruses as there are bacteria, fungi, protozoa, and archaea, which are primitive bacteria that live in the most extreme environments. The numbers are pretty staggering as well. Let's start with a ballpark estimate of over 100 trillion bacteria in the colon. It also represents, at a minimum, 100 times more genes than are contained in the human genome. Now there we go. I'd also mention that most scientists now recognize that the mass of microbes that's passing through us at any point in time is essentially an organ. The microbiota speak to one another in a chemical language and talk with the cells of your body, and your body can speak to your microbiota and affect its composition. Think about that. That's pretty amazing on something that's moving through you and is always in motion and is replenishing itself constantly. But it is thought of now as an organ system because of this crosstalk. Now, you're going to hear two terms, and you'll hear them frequently when you discuss this topic at all, one being microbiota and one being microbiome. Now, I've used these terms already, but I'd like to talk a bit more and lessen the confusion. Unfortunately, lessening the confusion probably won't happen quite yet, since scientists can't seem to agree on one definition. In my definition, which when I teach a class is the only definition that matters, that's my power in the classroom, is comprised of, the gut microbiota is comprised of the living microorganisms of the GI tract, the bacteria, archaea, protozoans, fungi, and the viruses that make their home there. They need us to survive, and we need them for our survival. The gut microbiome, as I define it, is comprised of the collective genomes of the gut microbiota. And as I stated before, it represents at a minimum 100 times more genes than contained in the genome of its host, the human. And as you can see from the slide in the red print, the microbiome has come to be used quite interchangeably with microbiota, but I believe they're quite different from one another. Nonetheless, you'll see them used interchangeably quite frequently. So are we humans alone in having a gut microbiota or microbiome? And the answer is simple: no, we are not. Every animal has a gut microbiome if it has a gut. Here's a few examples. Earthworms have a gut microbiota, and it probably is used in part to assist them in extracting all available nutrients from the soil they ingest. Mosquitoes have a gut microbiota. In fact, scientists are learning how to alter the mosquito gut microbiota with a bacterium that prevents the mosquito from transmitting the deadly dengue fever virus. Andean bears have a gut microbiota. In fact, I doubt you can think of an animal species whose gut microbiota have not been characterized within our ability to do so. The aquaculture industry is deeply involved with gut microbiota manipulation of fish in order to reduce disease and promote health. Pork and beef industries have been doing the same gut microbiota manipulations for years to maintain animal health and weight gain. And as with the relationship between the human host and the gut microbiota, the microbiota of all animals has established a symbiotic relationship with its animal host. Now, I mentioned earthworms, so let's go a step further with earthworms. Just for some perspective, it's important for us to understand the gut microbiota habitat. Humans are simply very complex earthworms. We're a collection of living cells making up a living organism that surrounds a hollow tube. We have a tunnel running through us from the mouth to the anus. The inside of the tube is, in reality, the external environment. Essentially, we have the same parts as the earthworm in our GI tract with a few more complexities. We don't seem to have a crop and a gizzard, although if this current pandemic goes on much further, I think my crop and gizzard are getting quite a bit larger as it progresses. And maybe I'm developing one. I don't know. Something's getting bigger in there. The concept's the same, however. Food goes in one end and waste out the other end. And in between, nutrients are removed for use by the worm, and the gut microbiota helps in that process. It also does a lot of other things, but in the case of the earthworm, nutrient extraction is a biggie. Now, the human intestinal tract, no labels on it, so it's just you're just looking at the raw thing here. Let's just start talking about its parts. We humans have a stomach wherein digestion begins in a very acidic environment. The pH of the stomach is 2 or less, usually less, and that's sufficient to kill off many, but not all of the microorganisms we ingest. Then comes the intestines. We'll call them the small intestines for now. The intestines are a series of pipes, about 22 to 25 feet in total length, maybe an inch, inch and a half in diameter. On the outside, the inside's quite a bit smaller. The small intestine is comprised of 3 sections: the short duodenum adjacent to the stomach, the jejunum, a 10-foot section where a lot of nutrient absorption occurs, and the ileum, another 10-foot section where the action begins to pick up microbiologically speaking, and more nutrient absorption continues. Now, at the junction of the small and the large intestine, known as the colon, is the cecum and the appendix. These are important structures, although they only recently gained status as the microbiota gained greater appreciation. The cecum is a pouch-like structure of the large intestine that provides the space for mixing the bacteria with the chyme being the partially digested foods that have moved down through the small intestine. In there, they undergo further digestion and fecal formation. The food microorganism mixture traveling through us is called chyme until it enters the colon, where it becomes feces. The appendix is a fleshy tube-like structure connected to the cecum, which is an essential structure for maintaining gut microflora and also in mucosal immunity, which we won't go into in a big way today. But the gut microflora, it's a place where the good bugs are stored. I guess we can think of it that way. The colon, this larger structure, is about 3 to 4 feet long and about 2.5 to 3 inches in diameter. And the colon is where the bulk of the gut microbiota reside. I mentioned before, 100 trillion bacteria reside in that structure. The colon is essentially a large fermentation vat where water and some nutrients are extracted and short-chain fatty acids are produced from fiber. And I'll talk more about the short-chain fatty acids a little bit later. The rectum and the anus are the discharge valves for the waste material or feces. Now, there isn't time to go into the intricacies of the intestinal lining structure, but let's just say that the many folds, now if this is a cross-section of the small intestine, it's just like you took a garden hose and sliced it. If you magnify that innermost layer, you see the folds. On the folds, you see the so-called villi, which add more surface area. And on the villi are the enterocytes, which are coated with microvilli. Again, magnifying the surface area for absorption. All these spaces provide unique ecological niches for the gut microbiota and expand the absorptive surface for nutrients to between 300 and 400 square feet. So it's quite an amazing architecture when you think about it. And finally, before we get into specifics, just a few final things about the gut microbiota, some generalities to just kind of carry along with you. The colon contains the highest microbial density recorded in any habitat on Earth. Did you know that about your colon? I didn't really until I started looking at this stuff. Between 300 and 1,000 different species, probably more towards the upper end. Besides bacteria, there are fungi, protists, archaea, and viruses. And I should also mention that some of us have a few helminths or roundworms kicking around as well. The number of viruses in your gut equals the number of bacteria, at least. So 100 trillion in the colon, 100 trillion viruses as well. Over 99% of the bacteria in the gut are anaerobes. They cannot tolerate oxygen. In the cecum, that blind pouch, the aerobic bacteria, ones that need oxygen, reach high densities. And many species in the gut have not been studied outside of their host because they cannot be cultured. So those are just a few facts to keep in mind as we go along. Now, about the distribution and abundance of bacteria in the human gastrointestinal tract, the number of bacteria in the stomach is very low. Again, it's suppressed by the acidic environment. As you go into the duodenum, it remains fairly low, around 100 per gram or milliliter of content. As you progress down the series of pipes, you go into the jejunum. At the upper end of the jejunum, it remains low, but as you go down the jejunum, the numbers start to increase, and then you go into the proximal ileum. This is still an aerobic because you're swallowing air through your mouth into the stomach and into the small intestine. The proximal ileum has a low number of bacteria, but most of them are aerobes or facultative anaerobes. They can do either way, either with or without oxygen. When you hit the distal ileum, you're up to fairly high numbers of bacteria. Now you're up in the billions, essentially, or close to 1 billion per gram of content. And you're getting into the anaerobic spectrum where there's very little oxygen available. And if there is any, it's being used up by the bacteria that are there. And then you enter the colon, which we've already mentioned. Now we're talking about billions, 1,000 billion, several hundred, or 100 trillion total. And it's all anaerobic. So it's a very interesting and also a very hostile environment. So when you talk about the microbiota, how do you talk about it? What's the language you use? And why do we give bacteria, archaea, and eukaryotes like ourselves? We are eukaryotes after all. Why do we give us names? Why do we have these fancy names? So the systemic classification of bacteria or other things on Earth refers to the naming of them and other organisms according to the binomial system, which was introduced by Carl Linnaeus in the late 1600s. And essentially it comes down to the fact that you need to provide names to give scientists, medical personnel, or anyone a frame of reference, a commonly understood way of communicating identity. I mean, if I just pointed at a bacteria under a microscope and said, look, a lump, and then another person would look in the microscope and say, well, I don't think that lump is what you're thinking it is. Well, that's kind of a crude way to try and communicate. But if you look and say that's Escherichia coli, everybody, microbiologists understand that. And the next microbiologist that looks through says, indeed, that's an E. coli, or you're not looking right, that's just a bubble. I don't know. But it gives people a frame of reference to talk about things. And before we go any further, just a reminder about microbiota versus microbiome. Again, according to me, and that's the only thing you have to go by right now, the gut microbiome is the totality of microorganisms and their collective genetic material present in the intestinal tract. The microbiota are the organisms of the intestinal tract, and this is where the names become important, those binomial names. So here's a binomial name. Proteus mirabilis comes from the Greek god of the sea, Proteus, and mirabilis, a wondrous, remarkable, amazing. That's where the word's derivation came from. And indeed, this is just one single organism that is truly wondrous, remarkable, and amazing. First of all, it's amazing because I got my master's degree working with this organism. My getting my master's degree was considered amazing by many people. So let's start there. But it truly is. If you spot, for example, just put one little dot of a culture of Proteus mirabilis in the center of a Petri plate with some nutrient material in that plate, and you wait a little while, the bacteria will grow from that spot, move outward, they'll migrate outward, and then they'll stop and rest for an hour or so. Then they'll start moving again, stop and rest. Then they'll move again. And they're going over a surface, a hard surface. And to do that, they form, these are the normal Proteus mirabilis cells, those little ones. They form these big swarmer cells loaded with flagella, which are the propulsion mechanism for bacteria. And that's the ones that migrate out and find this newer area. Now, the easy thing would be to say, well, they're just seeking more nutrients. Well, that's really not the case because there's plenty of nutrients still left in the plate right under them. So still haven't figured out after all these years. This was first seen literally at the turn of the century, the former century, and we still haven't figured out why they do this. So in other words, a lot to be learned about these critters. What about viruses? Okay, so these are the bacteriophage. Now, there are a lot of viruses in the gut microbiota, including viruses specific for humans, such as the enteric viruses or the enteroviruses. But the viruses that infect bacteria or bacteriophage are the most numerous. It's estimated that there's a 1-to-1 ratio of bacteriophage and bacteria in the gut. So 100 trillion bacteria in the colon, 100 trillion bacteriophage. Those, I'd hate to see the error bars on those numbers, but nonetheless, at least that puts you in some frame of reference. The bacteriophage infection of a bacterium usually results in the death of the bacterium. So bacteriophage are constantly attacking bacteria in the gut, destroying them. Bacteria of one species or genus are almost always attacking bacteria or species of another genus in the gut. And so there's this constant war going on. And it must look like the ultimate war zone. I imagine you could make a really good video game out of it. And yes, almost all the bacteriophage look like mini moonlanders. That's really what they look like. Bacteriophage have useful applications. They're used in and on many food types to eliminate pathogenic bacteria, such as E. coli 157H7 and Salmonella on meats and poultry. Phage directed against Listeria monocytogenes are used on some cold cut meats that have been sources of trouble in delicatessens. Phage are also now used as therapeutic agent for severe infections when antibiotics cannot be used. The US is quite late in adopting phage technologies as therapeutics, as the Russians have been using them since before World War II. It's believed that the ancient practice of bathing in the Ganges River to ward off illness and promote health arose from the bacteriophage in the Ganges that helped fight Vibrio cholera, the causative bacterium of the disease cholera. Phage are indeed plentiful in almost all waters tested, including sewage and ocean water, and pretty much any other water you come across. This is an electron micrograph of a bacterial cell, the large structure, being attacked by phage, and this poor bacterial cell never had a chance. So I'm using this as a transition slide because it shows a couple of things. First, it shows the epithelial layer of the intestinal tract, and let's say this is the distal jejunum. I'm sorry, distal ileum, and these are the absorptive enterocytes, and here are the microvilli, one of the things that expands the surface. And here are the bacteria and phage that float through on this stuff called either chyme or feces, depending where you're looking. So what I wanted to point out here is the gut bacteria. 90% are members of 2 phyla. Those are pretty big classification areas, and I'll show you those in a minute. Name the Firmicutes and the Bacteroidetes. These are the major gut phyla, and I'm going to talk a little more about these in a moment. Now, when I took bacterial systematics in graduate school, we didn't even concentrate on the phyla. Their importance and use has reemerged with the more recent interest in the gut microbiota in making some associations between the phyla and certain clinical conditions, which I'll just show you in one moment. But first, just to talk a little bit about classification, the Linnaean classification. This is one we can all relate to. In this case, we'll start with the phylum. I mean, the kingdom, they're all animals, okay? Phylum Chordata, a backbone. Mammalia, they're mammals. Carnivora, carnivorous. The family is Canidae, the genus is Canis, and the genus and species, Canis lupus, the gray wolf, from which all our domestic dogs have come. So that's the classification scheme for the dog. And in order to get equal time, we also have the systematics of cats. In this case, there are 2, Felis felis and Felis sylvestris. Felis felis, Felix the cat. Felis sylvestris, Sylvester the cat. For those of you old enough, suffer and succotash. So everything can be broken down by Linnaean systematics into the genus and species. And that's a handy way to refer to things so that whoever you're talking to knows exactly what you're talking about. Another thing that separates this big group of bacteria into smaller pieces, in this case, it's the simple staining procedure called the Gram stain that divides bacteria into 2 groups. If you go through the staining procedure, the blue stained bacteria are the Gram positives, and the red stained bacteria are the Gram negatives. And you can see the various shapes, some of the various shapes of bacteria. This is a light microscopic view of a mixture of bacteria, and you can see the rods, they're little dots. Some of them are pink, those are the Gram negatives, and some of them are dark blue, those are the Gram positives. Some of them are curved, those are vibrios, like Vibrio collar. So you could start to talk about these things by looking at them, classifying them, breaking them down into smaller groupings. Another breakdown is how they breathe, essentially. I talked about anaerobe and aerobe. Most of the bacteria in the gut are anaerobic. Both aerobic and anaerobic bacteria need to have an electron transport chain to synthesize ATP. This is just like human beings. We need the same thing. We have an electron transport chain in order to synthesize adenosine triphosphate, the molecule that provides energy. So while aerobic bacteria and humans use oxygen as the terminal electron acceptor, anaerobic bacteria use other substrates as the electron acceptor, such as sulfate, nitrate, carbon dioxide, iron, or even organic compounds. Oxygen is toxic to anaerobic bacteria. So that's why anaerobes, the colon, if oxygen doesn't get into the colon. If it did, it would kill off many of the bacteria that are living there. Now, one of the problems with studying anaerobes is they're more difficult to grow, whereas aerobic bacteria can grow in Petri plates or in tubes right on the countertop in your laboratory or in an incubator with plenty of air around. Not so with anaerobes. With anaerobes, you need to exclude the oxygen. In order to do that, you use things such as an anaerobic jar, which is a very tight sealed jar. Here are your Petri plates containing your bacteria that you want to grow. This is a hydrogen producing gas pack. You open it up, throw it in there, add some water. Hydrogen's produced, there's a catalyst up here. Any of the oxygen reacts with the hydrogen to form water, and the oxygen is slowly removed, and you're left with an anaerobic environment, no air. You can also do the same thing if you can displace enough of the air with carbon dioxide in like a large incubator. This is a glove box, and you can manipulate the things you're trying to grow. But they're hard to grow. So not many people have done these studies, quite frankly. And that's one of the reasons that our knowledge of what's going on in the intestine has been somewhat hampered. Now, for decades, classification of bacteria, saying what they were, was done by determining what a bacterium could do chemically. That is, what enzymes did it make and use. Sugars and the ability of bacteria to digest or utilize or break down sugars was one of the main distinguishing features. So you'd have a list of about 20 to 30 different sugar molecules, and you'd have little pluses and minuses. Could the bacterium break it down or could it not? And based on that pattern, you could discriminate between a lot of the different genera of bacteria. Now, we've come to a different point in time. Most of the classification is done genetically, not necessarily by needlepoint, but genetically. We use a highly evolutionarily conserved sequence of the bacterial ribosome. Now, if you recall from biology, the ribosome is an RNA structure on which protein is made from by transcription of the messenger RNA. And that structure is, again, highly conserved throughout all the animal species, except for certain very variable regions within the molecule. And those variable regions vary in their composition or their sequence. And that's what we can differentiate bacteria by using those variable sequences. Jennifer Glass must have had infinite patience to actually make this needlepoint. This is a map of the 16S, what they call the 16 Svedberg rRNA molecule. It has about 1,540 bases. That's AUGC, 4 bases, and 9 highly conserved or highly variable regions within the highly conserved molecule. Also note that all these little twists and turns in this thing are to show various folding points, because the ribosome is a very complex three-dimensional structure. It's not just a flat molecule, it's very tightly bound almost into an oval shape. So, and within it are these variable regions. By knowing those variable region sequences, you can take it down, take the systematic classification of a bacterium down to not just the genus and species, but even to the subspecies level sometimes. Okay, so within the human gut, there are 4 major phyla. We're only really going to talk about 2 of them, but these are the 4. Proteobacteria, Firmicutes, Actinobacteria, and Bacteroidetes. We're going to talk about the Firmicutes and the Bacteroidetes. I still stumble over these things after all these years. So first, let's ask the question, is there a core microbiota? In other words, do all humans have kind of the same structure of bacterial species in their guts at any point in time? And the answer is kind of complicated in a way, but not so much if you really just think about it for a minute. And the answer is that the taxonomic profile, that is the genus and species in humans, shows greater diversity than the repertoire of microbial genes present between individuals. So let's break that down. It means that, again, the genus and species, let's say I have an Escherichia Dougii, and you have an Escherichia Wendii. It doesn't matter that we're different in a way. It shows diversity between us, but the microbial genes present in those two bacterial species are probably doing the same thing. So the core microbiota, in a sense, might better be defined at a functional rather than the organismal level. In other words, it doesn't matter who is doing a critical job as much as it matters that the job is getting done. So as long as a gene is there that can do whatever that gene's supposed to do, it doesn't matter what species it's in as long as the job is getting done by one of those species. So let's look at something called the average human being. There really isn't an average human being, but the people make an attempt to come up with it by looking at the bacterial profiles or the phyla profiles of a whole bunch of people. And this is what they come up with. This big piece of pie are the Firmicutes, this piece of pie is the Bacteroidetes, and the rest are the others. And they always throw in a couple new names just to throw you off. That's what scientists do. But this is what they consider an average-looking person. Now, let's look at a term right now because it's an important term. In a healthy average person, the gut microbiota is balanced such that all normal physiological processes maintain health. So let's go back to this. So this balance of bugs is supposed to essentially maintain your health. Since you're average, you're not suffering from some dread disease, presumably. This is what's kind of keeping you going on a gut level. This is making you healthy. Dysbiosis, also called dysbacteriosis, is a term for microbial imbalance or maladaptation on or inside the body, such as an impaired microbiota. In other words, when the balance gets out of whack, when those pieces of pie start looking a lot differently, you have something called dysbiosis. Something's not in balance, and that's when problems can occur. So let's look at non-obese versus obese people. Now, in this case, this was a population of Japanese individuals that were studied. And you'll notice that in the non-obese people, the Bacteroidetes forms a pretty big piece of pie. Firmicutes, almost half of the pie. With obese people, Firmicutes is still about half the pie, but the Bacteroidetes has shrunk. I have no idea what these unclassifieds were. They didn't take them much further than that, so they were just a bunch of bacteria that they had a hard time identifying. But these are the two phyla we want to focus on, Firmicutes and the Bacteroidetes. And what the conclusion of this study was that the Firmicutes to Bacteroidetes ratio, as it increased, as the Bacteroidetes decreased, the Firmicutes to Bacteroidetes ratio increased, and so did the body mass index. So there was some correlation that they noticed between the shapes of those pie charts and whether or not someone was going to be or was obese versus not obese. What about another correlation? Let's look at the influence of dietary components. Looking at breastfed versus formula-fed infants. Breastfed infants, this huge chunk of proteobacteria versus the formula-fed. Except I swore I wasn't going to talk about proteobacteria, but I just went back on my word, I guess. Formula-fed had more Firmicutes. And the amazing thing no Bacteroidetes in the formula-fed infants. So it was pretty easy looking at the phyla profile between just breastfed and formula-fed infants to say, wow, there's a difference, and probably a predictable difference. Even elderly versus young. If you look at elderly subjects versus younger subjects, two things will stick out. Number one, the elderly subjects. Again, the Bacteroidetes, the increasing number of Bacteroidetes. The other thing is that you can't see in this is that elderly subjects have a much diminished diversity. There are fewer genus and species in total number than there are in younger subjects. So when you age, you lose diversity, and you also seem to lose your Firmicutes, which are thought of as a good group of bugs to keep around. Trust me, folks, there's nothing good about getting old. Okay, now what I want you to look at and think about for a second is a cross-section of the intestine. Remember when I sliced the garden hose before? This is the same thing, except here it correlates to what you'd be looking at microscopically. So you're looking at the epithelial lining, the microvilli. You're looking at a mucus lining, which is simply called the inner layer and outer layer of mucus. The inner layer is a tightly bound layer, and there are very few bacteria in that layer. And the outer layer is the loose layer of mucus that contains large number of bacteria, and it's always moving. It's being propelled slowly towards the anus because of peristalsis. And that's what these rings depict. This white area is the lumen. That's the tunnel that goes through you or goes through the earthworm. Here's another cross section just to remind you that within that lumen or the tunnel is the waste material moving through you at all times. And that waste material contains the trillions of bacteria that we're talking about, and it's also the organ that we're talking about. So here, this is kind of amazing. You have an organ moving through you at all times, and it's not really part of your body. Your body just surrounds it. Kind of weird stuff to think about, but it's true. And here, the luminal bacteria. The waste material contains high populations of Bacteroidetes. There's an interface between the outer loose mucus layer, which is populated mainly with Firmicutes. And this interface is an area known as the area of cross-feeding. Now, what do I mean by that? This is where we start to get into the nutrition stuff, which is the fun stuff, really. All right, let's take a complex carbohydrate such as a fiber that's moving through your digestive system, and it finally gets to the colon. There's a group of bacteria in your colon known as the primary degraders. These are bacteria like bifidobacteria, lactobacilli, Bacteroides, not Bacteroidetes, Bacteroides. And these bacteria possess enzymes that can degrade these complex carbohydrates. They can break them down, and after further metabolism, form acetate and lactate, very simple molecules. There are also bacteria in the colon that can take acetate and lactate and produce butyrate. And these are some of the genera that can do that: Rosburgia, Eubacterium, Clostridium, Ruminococci. They have no ability to degrade these complex carbohydrates, but they can take the acetate and lactate from the primary degraders and produce butyrate. Now, the primary degraders, bifidobacteria and lactobacilli, you've probably seen those words on the labels of most probiotics that you pick up in the grocery store. So primary degraders are, in fact, probiotic organisms, the ones that can break down the complex carbohydrates. But in order to get to a useful end product, or a really useful end product, butyrate needs to be produced, and you also need these bugs in your gut, and these are not in the form, usually, and if they are, they're probably not legal, of any kind of a commercially available probiotic. So you got to have them all. The primary degraders you can buy off the shelf. Butyrate producers, not so much. To give you an idea how important butyrate is, There are two species of bacteria, Faecalibacterium prausnitzii and Eubacterium rectala. These are important butyrate producers, and they constitute up to 12 to 14% of the total gut microbiota in fecal samples of healthy adults. Now think about that. Two species comprise 12 to 14% when there are 300 to 1,000 species present. That tells me that butyrate must be pretty darn important if it's there, if the producers are there in that high ratio. There are lots of other species that produce butyrate, butyrate in the gut. So these are the other ones. These are the some of the other butyrate producers that take the products of the primary producers of the probiotic and translate it into butyrate, which is one of the three major short-chain fatty acids produced in the gut, the others being acetate and propionate. All three of them are probably important. Some of the things that butyrate does, first of all, it's an energy source for intestinal epithelial cells. In fact, the colonocytes, the colonic epithelial cells, depend almost entirely on butyrate for their energy. It can be used to treat inflammatory bowel diseases. It's a possible therapeutic agent for multiple sclerosis and neural autoimmune diseases. Butyrate inhibits mast cell activation. In other words, suppresses the effects of allergic reactions. It also, by inhibiting mast cell activation, probably has a beneficial effect in the lung problems associated with COVID-19. It's a treatment for multiple myeloma, acute leukemia, and acute myeloid leukemia. And it's an anti-inflammatory both in the gut and systemically. So it's a pretty amazing molecule that we're just starting to appreciate all the things it does. But I don't want you to forget that there are hundreds of molecules produced by the bacteria going through this magic new organ that's traveling through you that are interacting with other cells in your body and doing miraculous things. To give you an example, this is from Dysbiosis. This is just a summary of some of the things that have been associated with dysbiotic gut microbiome. Autism, Alzheimer's, psychosis, arthritis, hypertension, allergies, asthma, leaky gut, heart problems, and intestinal problems like Crohn's disease and inflammatory bowel syndrome. So the dysbiotic gut is becoming more and more associated with almost of maladies of almost all the major organ systems, coronary being a very, very serious one. So, you know, it's important stuff. And we're just, again, we're about where we are when astronomy launched the Hubble telescope. This just sums it up again. There are connections between the gut and other major systems. There's a connection from the gut directly to the brain. And all of these associations are being noticed. You know, you are what you eat, maybe, because you can get depressed, you suffer anxiety, it can trigger their thinking, it can trigger autism, multiple sclerosis. If this gut microbiota becomes too dysbiotic, or this gut barrier, this single epithelial layer, becomes too leaky. And finally, just keep in mind, your gut bacteria don't care so much what you call them as long as you feed them. And thank you for your attention.
Well, thank you very much, Dr. Archer. I think that was a really great summary of the background for things to know as we go on with the series. Wendy Dahl was going to talk a little bit about diet and microbiota, but we're going to move that to our November session. That'll fit in. Next time we're going to talk about probiotics. So first of all, it's a great presentation. I always learn so much on these. And so with that, I think we'll just go directly to questions. So Kendra will be reading off our questions. I know there's a lot of stuff, a lot of great questions coming up.
Yes, there were a lot of really great questions. So Dr. Archer, there were a couple questions related to the appendix. Are there studies to suggest that when the appendix is removed, a person experiences dysbiosis?
Well, right after it's removed, you certainly do. In fact, you're dysbiotic when you go into the hospital. Um, Going into the hospital is a pretty traumatic thing, and your diet's probably been upset before you get in there, and a bad diet can trigger dysbiosis. So when you're in the hospital, most people don't like to eat hospital food. Um, Once you cut off your fiber source, for example, your gut immediately responds by not producing, or doesn't have the fuel to produce the acetate and lactate through the probiotics and uh, work its way down through the butyrate. So things get very messed up very quickly. But that's any surgery. It wouldn't just be the appendix. More to the point with the appendix is the loss of an immune organ. Um, with many immune organs, however, enough cells are dislodged and go to other parts of the body and essentially set up mini appendices elsewhere. So it's possible you don't lose much. Same thing happens with tonsils. So it may well happen with the appendix. The appendix, in terms of being a storage place for important bacterial species, that's a rather new concept. And certainly people live very nice, long lives without an appendix. So somewhere else, the body makes up for that slack.
Okay, I think my microphone wasn't working, so can you hear me a little better now?
It is
Yep.
better.
Excellent. Um, so someone who has an appendectomy, do they struggle to produce flora versus someone who hasn't?
It doesn't seem so. I think there are backup systems um, in the human body where, perhaps the cecum takes up the slack, or perhaps the appendix, even though it looks like it's removed, what you're really removing is the necrotic, or the area where the infection is. There are still cells of the so-called appendix there as present. And there are other ceca, there are other little pouches in the gut. The appendix just happens to be one that is big. And perhaps the other little ceca take up the same role. I don't know. I don't know if that's really been studied that much. It probably has, but it would probably be out of my area. As I said, I don't think there's anything to argue that people without an appendix are any worse off than people with one. I think they would avoid taking it out so long as they could. But once an active infection of the appendix is going on, which is sort of, it can be a very rapidly progressing infection to cause sepsis, they have to take it out.
Okay, next couple questions relate to the colon. So given the large bacteria population in the colon, what might the consequences to microbiota or one's health in a general colonoscopy prep or purge?
Wow, there's so many. I mean, a colonoscopy, my understanding, I'm not a physician, but it's something that you don't want to volunteer for. They can be miserable. There are numerous opportunities for sepsis to occur where bacteria get into the peritoneum. It can be a dangerous procedure. People with Crohn's disease, for example, I'm sure, or if anyone of you listening, I hope you don't, but if you do have something like Crohn's disease, you're probably well aware of what it can do. It wouldn't be a fun thing. I don't know how else to say it. I would rather not go any further. Again, I'm not a physician, but a lot of bad things can happen. On the other hand, when you have to have a colectomy, it needs to be done. I mean, there's not like you have an option. So and they don't just say, well, gee, let's lop off a piece of your colon. I mean, it's just not done lightly. It's done to save your life usually. So you haven't got much choice, but it's not a fun thing to have.
Okay, I think that addressed what if the colon is removed, right? So what about the colonoscopy, like the procedure where
Oh,
the
okay.
other procedure,
The colonoscopy?
right? Like for the screen, isn't that the screening for the cancer where you have to purge your colon to go in with the scope kind of thing?
Yep. You never really purge it. I mean, that's the thing about bacteria. I think a lot of people are learning that now from the analogous situation with what we're learning about viruses because of the current situation. You know, you can get viruses on a surface and it's very hard to remove them. And the same things happens in the colon, either even harder to remove. So when you quote purge your colon, it doesn't mean you're removing all the bacteria. You're moving all of the feces, perhaps, or most of it, but there's still a lot of bacterium sticking around in those mucus layers. And so as soon as you start eating again and things start moving through, you just re-inoculate the chyme or the fecal matter as it goes through you, and you're right up back in business in terms of microbiology. So you never really get rid of your bugs. I don't think, I'm not aware of any way to sterilize something like the colon, but um, it'd be very hard to do. But certainly the prep for, you know, colectomy, no, it wouldn't do that. Or a colonoscopy, I'm sorry.
Okay, we had a number of questions about probiotics, and in the interest of time, we're going to hold those questions for our next session because that is going to cover probiotics. So I think we have time for one or two more questions. We'll be sure to get all of your questions answered. There were a lot of really great ones. But the next question was about antibiotics and how they affect the microbiota.
Wow, they can blow it apart. That's just sometimes, depending on the antibiotic, they can really mess up your gut and cause dysbiosis. They can trigger all kinds of problems like something called SIBO, small intestinal bacterial overgrowth, where the bugs from the colon sneak back up into the lower intestine and they explode in growth and can cause some real discomfort. So yeah, I mean, I could go into antibiotics right now and fill an hour, which I evidently have gone over time, and I apologize for that. And so I don't know how to answer this question quickly. But antibiotics, more and more, it's being shown that antibiotics, even like in infancy, can have permanent effects on an infant working via the microbiota in the gut. So yes, antibiotics have a dramatic effect on the gut microbiota. And that's why, in many cases, people take probiotics, adults take probiotics to restore some of the flora of the gut after you take the antibiotic and wipe it out. So when you take an antibiotic, you're killing the bad bugs, but you're also killing a lot of good bugs along with them.
So what is the best way to repopulate after you use an antibiotic?
Eating a good diet and eating things with fiber, eating things with live bacterial cultures like yogurts, fermented foods of any kind would help, and all those things restore. And I think most of you are probably aware that in most hospitals now, which has not always been the case, yogurt's kind of a staple food, and they've caught on that that's something that can help maintain a good, healthy gut flora if you're on antibiotics.
And then a question related to, I think when you were talking about infants and microbes and the study that you were talking about, can you talk about what age those infants were and how the microbes changed over time?
Well, those studies are very numerous, number one. I'm sure they've been done at multiple age ranges. Most that I have read were between 0 and 6 months. I mean, from birth to 6 months. But even later in childhood, neonate up to toddler, the same thing can happen. And, you know, the kids that were treated with, for example, with antibiotics early in life are more prone, for example, to allergy later in life. They've just lost something. Something has happened, a triggering event, and they are either more prone to a certain set of diseases or conditions such as allergies. And that can last for a long, long time. So all I would suggest to look in the literature, even Google search or PubMed, and look for the age range you'd be interested in, because it really is broken down by age range.
All right, well, this is Julie England. I think we're right at the 4 o'clock mark. So first of all, we want to thank Dr. Douglas Arthur. Too bad I can't pronounce his last name. Thank Dr. Archer for a really interesting subject topic today and answers to our questions. If for some reason we have more questions, non-probiotic, because we'll be talking about probiotic, you can always email one of our team members and we can get the answers for you. As I say, thank you again. And so next time in October, I forgot which date, we'll be talking about probiotics. And then following that, following week, we'll be doing diet, microbiota, and fermentation in November. And so anyway, thank you very much. And we appreciate you taking part in today.